In this segment, it discusses visual systems. So it starts out by talking about visual systems extends from the eye to the brain, and it talks about how the eye starts with the cornea and the lens to focus the light, and then the light passes straight through in a line until it's refracted by the cornea, and then the focus is adjusted by ciliary muscles inside of the eye that cause accommodation. Light the eye is then controlled by the pupil, the black part of your eye, and movement is controlled by three pairs of extraocular muscles that extend to the orbit. So visual processing begins in the retina, which is the receptive surface inside the back of the eye with several layers. And the photoreceptors detect light, and they come in the forms of rods, which are long and narrow, and cones, which are different and respond differently to light-varying wavelengths, providing us with color vision. Both rods and cone photoreceptors release neurotransmitter molecules that control the activity of the bipolar cells that synapse them. Bipolar cells connect with the ganglion cells that form the optic nerve. Horizontal cells and amacrine cells are significant in the lateral interactions within the retina. The horizontal makes contacts among the receptor cells and bipolar cells, and amacrine cells contact both the bipolar and the ganglion cells. All only create local graded potentials, and not action potentials, and these cells affect each other with the greater release of neurotransmitters in response to changes in electrical potentials. There are approximately 100 million rods, 4 million cones, and 1 million ganglion, and that's because the ganglion are larger and much more energy costly. Two different types of functional systems are scotopic and photopic. And scotopic use rods and work in dim light and have lots of conversions on the rods to the ganglion. And photopic use cones that require light and differential sensitivity to wavelengths and have less conversion. And then the photoreceptors transduce light into chemical reactions. So only a fraction of the light that strikes the cornea actually reaches the retina because of so much of the convergence and how the light passes through. And then it talks about rhodospin, which is the photopigment in rods. And the photopigments in the eye consist of retinal and opsin. And retinal refers to a molecule, not actually referring to the retina. They are visual receptor molecules that span the membrane of receptor discs and have structures that are similar to those of G-protein coupled neurotransmitter receptors. So when rhodopsin is struck by light, retinal dissociates rapidly from the opsin molecules to reveal the enzymatic site. And then the altered opsin molecule rapidly activates many molecules of the G-protein transduction, breaking apart the cyclic GMP and sodium channels open to stimulate light which the stimulation by light initiates events that close the channels afterward. A single quantum of light can close hundreds of sodium channels by blocking entry of more than 1 million sodium ions into a hyperpolarizing potential. Size of the hyperpolarizing photoreceptor potential determines how much less synaptic transmitter will be released, and thus release less neurotransmitter. Increase or decrease in the intensity of light can stimulate the visual system, and hyperpolarization is just as much a neural signal as depolarization. The visual receptors required help account for three major characteristics of visual systems, which are sensitivity, because weak stimuli are amplified to produce physiological effects; integration of the stimulus over time, which makes vision relatively slow but increases its sensitivity; and adaptation of the visual system to a wide range of light intensities. Different mechanisms enable the eyes to work over a wide range of light intensities. An adjustment of pupil size allows the eye to absorb numerous light ranges that are controlled by specialized retinal ganglion. Another mechanism is range fractionation, and it's when a wide range of intensity values can be encoded by a group of cells, each of which is a specialist of the particular range of the stimulus intensities, and no additional refraction can be done without vision issues. Every photoreceptor has a range of adaptation or progressive loss of receptor sensitivity as stimulation is maintained, and it's the main reason why we can't see over wide ranges of light. Two main factors that account for this are the varying calcium concentrations and the recombination time of retinal and opsin. Acuity is best in foveal vision. The visual field is the area that we can see without moving our eyes or our head, so without moving. Okay, and the fovea is the central region of the visual field where acuity is best because it's dense with cones, and acuity is the sharpness of vision. The optic disc is near the nasal side of the fovea, where blood vessels and ganglion cells and the axon leave the eye, and there's no photoreceptors there, and it causes a blind spot that you usually do not notice. Cones and rods are larger towards the periphery, which is the sides, which is what your peripheral vision comes from. Rods are distributed differently than cones, and they're completely absent in the fovea, but more numerous in the periphery. And that's why acuity is decreased due to increased convergence on the rods, because they go to the ganglion cells. Brightness of things is created by visual systems, usually by lateral inhibition, which is a phenomenon by which interconnected neurons inhibit their neighbors to produce contrast. So this is often seen by covering one side of a stripe and seeing how those different stripes affect how you see the specific one. Neural signals travel from the retina to several brain regions, and it goes from the ganglions to the axons to the optic nerve to each side of the brain to visual cortex and the occipital lobe, which is in the back of your head. The optic chiasm is where the optic nerves cross the midline, and axons from half the retina toward the nose cross the opposite side of the brain, and half of the retina toward the side of the head project its axon to its own side, which broadens the visual field. The optic tract is when the axons of the retinal ganglion after the optic chiasm that normally terminate on the lateral geniculate nucleus, leading to the optic radiations that terminate in the primary visual cortex of the ostriate cortex. The retina projects to the brain in topographic fashion, or 2D vision devoted mostly to the fovea, and we can predict certain types of blindness because of this due to injury by looking at the map of the 2D field. Neurons at different levels of visual systems have very different receptive fields, and photoreceptors excite some retinal neurons and inhibit others. After hyperpolarization, less glutamate is released and depolarizes one group but hyperpolarizes another. On center bipolar cells, seeing light in the center excites it, releasing less glutamate, which would normally inhibit the bipolar cells. Off-center bipolar cells, turning off the light excites them because it receives more glutamate, depolarizing the off-center bipolar cells. Bipolar cells also release glutamate, and glutamate always depolarizes ganglion. Neurons in the retina and LGN have concentric receptive fields. Cells around one another have opposite effect on their neighboring cells. So one will be inhibitory, one will be positive, one will be excitatory, one inhibitory, one excitatory. And the two layers of the receptive fields are on-center off-surround or off-center on-surround. All right. So the lateral ganglion. Sorry, I lost my spot. So the lateral ganglion has 6 layers structured called the geniculate, and there's 4 outer layers that are the parvocellular, 2 interventral layers that are the magnocellular, and these neurons can discriminate wavelength. The neurons in visual cortex have varied and complicated receptive fields. Light for the LGN and the ganglion are not very effective for cortical cells. And cortical cells come in simple or complex, and some require movement. Simple respond best to an edge or bar that has particular width, orientation, and location in the visual field, so things with exact size. Complex, elongated receptive fields, but also respond to size and orientation anywhere within the larger visual field. The spatial frequency filter model is a model of pattern analysis that emphasizes the Fourier analysis, and is typical with the number of light and dark cycles that stimulus shows per degree of visual space. That's it. Thank you.